Alright, in these two assignments, the pairwise and the multiple alignments, you're going to have to go to websites and you're going to use the tools there. These are called alignment tools. Pairwise alignments, alright, I'm going to copy this right here, this version. I'm going to go to that site. And pairwise alignments are very similar to dot plots, except for you're getting the sequences instead of the a visual, which is the dot plot. You're actually getting the sequences, and it's telling you which sequences are aligning or where they're the same. Now you're going to have two types. You're going to have global alignments and you have local alignments. And so you're going to have to run your files that I tell you to do pairwise. You're going to run them in a global alignment. So you'd use nucleotide here. You would go here and you would paste in your two sequences just like you do with dot plot. Alright, and you submit, and it's going to give you an answer, and you're going to have to interpret that. So you're going to do that one, then you're going to do this one, and these are going to be global alignments, and some of these are going to give you different answers. Then there's going to be local alignment, and there are 3 programs to run for this one. So of these 1, 2, 3, 4, 5 programs, you're going to essentially you're going to paste your sequences in or upload them to the program. It's going to give you an answer, and you're going to have to interpret the results from these 5 programs and tell me which one you think is aligning them better. Some of them may be better at aligning certain parts of the sequences to each other. Others may not. Some of them aren't going to do a very good job at all. You have to tell me which of these is best for the sequence I gave you. Now that's for pairwise alignment, so there are only going to be 2 files you put in for that. For the other one, this is multiple alignment. Now in a multiple alignment you put in multiple files, but I have created for you a file called the alignment file, and it already has the sequences pasted into a single FASTA. So when you go to the programs, and in this one there's just one type, there aren't the global and local, it's just one type here, but there are 7 different programs here, and so you'll have to run this 7 times and look at the results. And here, this is set for proteins, we're going to do nucleotides, so DNA or RNA depending on what you have, and you would just upload the single file that I've given you that has multiple sequences in it. You shouldn't have to change any of these options, and you click submit and it will give you results, and you will have to interpret those. But there are 7 different programs for this one. Alright, now these are worth more points. They're like 20 or 30 points for doing these, so they're worth more. They do take a while. I've given you 2 weeks to turn these in. So go and try these again. If you have questions on what you're supposed to be doing, post it, and I will answer as well as I can, or I'll make another video that will tell you a little bit more about it.